Hi there. So today I'd like to talk about this drug called OnabotulinumtoxinA, and specifically its use in cosmetics. You might be wondering what this actually is, but I'm sure you've heard about its generic name, Botox. And on the left here is a screenshot from my current favorite show, Selling Sunset. It's just to show you how much people love Botox right now. And whether you're just curious about Botox or at some point you're considering getting a Botox, I will be presenting some scientific data on it, and you can kind of weigh it out and see if Botox actually worth the hype. So let's get into it. First of all, what actually is Botox? So it is a drug made from a toxin, and that is produced by this specific strand of bacteria called Clostridium botulinum. So the structure of this protein is shown at the bottom right. It consists of a heavy chain and a light chain and are connected by a disulfide bond. Together, they are 150 kilodalton heavy. So it is an acetylcholine release inhibitor and a neuromuscular blocking agent. It was first approved by the FDA in 1989 to treat eye muscle disorders, and then it was approved for the treatment of cervical dystonia in the 2000s. And as for Botox Cosmetic, it was approved in 2002 for, and I quote, the temporary improvement in the appearance of moderate to severe globular lines associated with corrugator and or precarious muscle activity in adults 65 years and younger. So there are two things I think worth noting. First is that it's only a temporary improvement. And then second of all, the FDA only acknowledged its use to treat globular lines, which is the wrinkles on your forehead that is caused by frowning. But of course, currently people are using it more than just on their foreheads. They are also using it around their eyes to treat the so-called cross feet. But this is just something to worth noting. And Botox Cosmetic actually contain the same active ingredient in the same formulation, but just with different labeled indications and usage. So I want to briefly talk about the mechanism of action. I mentioned earlier that it is an acetylcholine release inhibitor, and acetylcholine is a neurotransmitter of the parasympathetic nervous system. So on this diagram on the right here, this is acetylcholine, and after being transported in a vesicle, it can be released to the neuromuscular junction, and then it can get to the postsynaptic cells through receptors. It is very important for contracting smooth muscles, dilating blood vessels, increasing bodily secretions, and slowing down heart rate. So what Botox does is to basically inhibit the release of acetylcholine at presynaptic cell. So there are four steps. First is the receptor binding domain of Botox on its heavy chain to bind to the receptor on the surface of presynaptic cells. And then Botox is internalized by forming a vesicle. Then it is translocated into the cytoplasm of the nerve terminal using its translocation domain on the heavy chain. Lastly, the catalytic domain inhibit the release of acetylcholine by cleaving this protein called SNAP25. So Botox is a protein, so the unit for dosage is a little bit different. It's actually just measured in an arbitrary unit. So that unit, it's defined as the LD50 in mice, the medium lethal dose. And one unit is approximately 0.05 nanograms. And it was first tested in monkeys in 1973. In monkeys, it showed reversible muscle paralysis started from day 1 to day 15 after injection, and that lasted for 3 to 4 months as the new neuromuscular junctions are formed. And in monkeys, the LD50 is 39 units per kilograms. And the LD100 is actually very close to LD50, which is around 41 units per kilogram. And an interesting thing is that in mice, the LD100 is 2.2 times the LD50. So that shows that mice model might not be such a good prediction for primates. But again, we don't know how good the monkey model is for humans because there's no ethical ways for us to test that. And there is no animal studies or phase 1, phase 2 clinical studies that are specifically designed for its cosmetic use. As for the animal study, I presume it's because it would be really hard to evaluate the muscle smoothing effects in mice or in monkeys. But how it got started to use as a cosmetics is that 2 Canadian dermatologists just started to use Botox on their patients for facial wrinkles. And they got really good results and published their results in 2 papers, one in 1992 and another in 1994. And since then, people just started to use Botox for cosmetics. And as I mentioned earlier, it wasn't until 2002 that FDA officially approved its use for cosmetics. And here I want to present a clinical study, one of the 2 clinical studies that got Botox cosmetic to be approved by the FDA. So it is a phase 3 clinical trial. It is a double-blinded, multi-center, and it was randomized based on age groups. So there were 264 patients in total, in which 203 were assigned to Botox treatment and 61 were assigned to placebo. All of the patients were between 18 and 75 years old with globular lines of at least moderate severity and maximum frown. So these patients were asked to frown as hard as they could without closing their eyes. And then the physicians would rate their severity of their globular lines based off of a photo scales. And that is on a scale from 0 to 3, with 0 being none and 3 being severe. So all of the candidates in this study are 2 or 3 in the rating system. And on the right here shows how the injection worked. So there were 5 injection points on the foreheads. Each injection was 0.1 milliliter, which is 4 units for the Botox groups. And in total, that is 0.5 milliliters and 20 units for the Botox group. And here are the results of this study. So on the left here, the 2 graphs shows the results of patients at maximum frown, and they also assessed patients' globular line at rest. So on the top 2 graphs here, it shows the mean score of patients' globular lines. So the black bars show the Botox group and the white bars show the placebo group. And you can see that starting from day 7, the Botox group started to see a very dramatic decline in the mean score, and the lowest achieved was at about 30 days. And then similarly at rest, the Botox group achieved a much lower mean score, and the lowest was at about 30 to 60 days. And then the bottom 2 graphs here show the percent responders, which is the percentage of patients who started out had a rating of moderate or severe globular lines and then changed to mild or none. So for the Botox group, this is starting from day 7, over 80% of the people showed a response. And there were barely any changes in the placebo group. And similarly at rest, the Botox group achieved a much higher percent responders, whereas the placebo group stayed at between 20 to 40%. And now, as for the adverse events, there was no serious adverse events that could be linked to Botox. And some of the most frequently reported adverse events were headache, respiratory tract infection, blepharoptosis, back pain, and acne. Of these 5 adverse events, only this one, the blepharoptosis of the Botox group, is a lot higher than the placebo group. So this is when the patients cannot fully open their eyes. Their upper eyelid could not come up fully. Which makes sense since Botox is a neurotoxin and the injection points are very close to their eyes. So it is possible that the toxin could travel and numbing the muscles around the eyes. And then another study that I want to talk about is after Botox Cosmetic got approved, there was a study that focused on dosage ranging long-term effects of Botox. So they used different total injection volumes of Botox. They started out at 10 units and then raised to 40 units. So this graph on the left here shows the percentage of subjects rating mild or none at maximum from after the treatment. And they traced it all the way through 10 months after the injection. So the only line that is a bit off from the rest is this one that represents the 10 unit Botox group. So that this means that the patient's wrinkles came back faster than the other 3 groups. And then similarly, on the graph on the right here shows the percentage of subjects returned to baseline for 2 consecutive visits for both at maximum front and at rest. And again, the 10 unit Botox group, they recovered to baseline much faster than the other 3 groups. And for all 4 groups, the effects of Botox basically weared off at about 6 to 8 months. So that means 10 units of Botox is probably not enough for a longer lasting effect of wrinkle smoothing. And that 20, 30, and 40 units don't show too much of a difference. So 20 units is usually the sweet spot that people use. And then finally, there is also a possibility of immunogenicity, which is the emergence of resistance to Botox as a result of formation of blocking or neutralizing antibodies. So there was a meta-analysis done using 16 clinical studies with a total of 2,240 patients who meet the criteria for the study. And something worth noting is that this study was sponsored by Allergan, the company that sells Botox. But this chart over here presents the number and percentage of people started out being antibody negative and then turned antibody positive after 1 or more treatments. So the most So the most frequent immunogenicity is the treatment of cervical dystonia, in which 1.28% of the patients showed this effect. For the other 4 usage of Botox, the immunogenicity effect was all below 0.5%. So that study concluded that it is unlikely for Botox to have immunogenicity effects, but because there is a conflict of interest in this study, I would suggest to take the result with a grain of salt. And that is all I'd like to talk about it today. And I hope you've learned something interesting, and thank you for listening.